In this lecture, we are going to talk biotechnology, looking at the medical and industrial applications of genetics, which were the subject of our previous lecture. So here's the thing about biotechnology. Biotechnology is a huge, huge venture that involves so many different kinds of genetic applications that really it would be difficult for us to talk about all of them in a single lecture. In fact, you can get a degree in biotechnology. You can take courses that devote themselves to different aspects of biotechnology. So what we're going to do here is try to apply the concepts that we learned in our genetics lecture to certain aspects of biotechnology. And so what we're really going to talk a lot about is using recombinant cells, which, as you remember from the previous lecture, are recombinant because they have DNA inside of them that has been donated to them from an outside source. So that was something we talked about in the previous lecture. We talked about horizontal gene transfer as a natural phenomenon. Today, what we're going to do is talk about horizontal gene transfer as an industrial phenomenon, an in vitro phenomenon, where we are preferentially inserting DNA into recipient cells in order to recombine them in such a way that they can have some therapeutic application. Those applications are very wide-ranging, and they might include the in vitro production of human enzymes or other proteins. They might include the production of subunit vaccines or the experimental viral DNA vaccines that you might be hearing about because of the COVID vaccine. They include gene therapy applications, disease identification applications, and they all have in common that at their heart there is a gene of interest, as it's called, that has been cloned or copied from a donor organism and then is placed into a recipient cell in order to express that piece of DNA. And so what we call all of that together is recombinant DNA technology. Recombinant DNA technology is the use of in vitro produced recombinant cells in order to make some therapeutic or industrial product. If you're interested in some of the pharmaceutical products that are produced as a result of recombinant DNA technology, there's a table in the lecture notes that you can access. What we're looking at here is just a very basic overview of this process of creating recombinant cells. What we're going to do after we look at this very quick overview is go and look at each of the constituent parts and see what microbes have played a role not only in producing these pharmaceutical or industrial products, but in making the cells that are able to produce these pharmaceutical or industrial products. So it begins with, in this case, a human cell which has a gene of interest that is insulin. This is an incredibly important pharmaceutical product throughout the entire world for all people who have insulin-dependent diabetes. And prior to the recombinant microorganisms making insulin, it was necessary for insulin to be harvested from the pancreas of slaughtered pigs, which led to the possibility of zoonotic or animal transferred diseases. It also produced an insulin that was not quantified as carefully. So this revolution in the treatment of insulin-dependent diabetes, which begins with bacteria or yeast cells producing insulin, has really revolutionized the way that diabetics can be treated and has done a ton to increase both the longevity and the quality of the lives of insulin-dependent diabetics. So insulin here is our gene of interest. Insulin is the thing that we want microorganisms to make for us. So we need to cut that gene out of the human genome, and we need to insert that DNA into a bacterial cell. Now, we know that there's a process of transformation, and transformation can occur when a piece of DNA is found in solution and taken up by bacteria. That's a very uncommon process, and as you might remember from our previous lecture, it relies on the bacterial cell being able to use and transcribe and translate that protein, and it also is necessary that that cell is able to pass on that protein to its offspring. And the easiest way for that to happen is for that gene of interest to go into the cell as a plasmid, because a plasmid is a self-contained piece of DNA which can be transcribed and translated into proteins and can also be replicated. So you've got to find a way to get your gene of interest into a plasmid, and then you've got to take that recombinant DNA plasmid, right? That plasmid itself is recombined because it has the plasmid DNA shown here in blue and the human DNA shown here in red. And then you have to get that DNA into the bacterial cell, creating that recombinant cell, which will then transcribe and translate the human insulin. And then you've got to collect that insulin in enough quantity that can actually be commercially useful to you. So that's the overall process. The question is, how do we make this happen? And the first issue that we have is, how do we cut a gene out of a chromosome? How do we get just a single piece of DNA out of a chromosome? And then how do we cut a plasmid such that we can get that other piece of DNA into the plasmid, creating the recombinant DNA molecule? So what you need is a really, really small pair of scissors. In 1976, those little tiny molecular scissors were discovered, and they were given the name restriction enzymes. And they were named restriction enzymes or restriction endonucleases because they could restrict or cut a nucleic acid. And they were discovered in bacteria, and they actually comprise part of the bacterial defense system. It's sort of like a bacterial immune system, where if invading viral DNA from a bacteriophage enters the cell, these restriction enzymes will become activated and they will go chop up the viral DNA. Then it can't direct the synthesis of new viruses, and so the bacterial cell is safe from the end result of viral infection, which is lysis of the cell. What researchers discovered was that those molecular scissors could be used to cut or restrict a piece of DNA in vitro, in the lab. So you see here a table with 4 different restriction enzymes. I'm going to point out just the second one, and we'll focus on him for a while, and that is a restriction enzyme that was discovered in Escherichia coli and was given the name EcoR1. Notice eco in italics to indicate to you that that's from its genus and species name, and R1, a designation indicating that this was the first restriction enzyme discovered in E. coli. And these restriction enzymes don't just cut DNA at any site. They actually have what they call a recognition sequence, which for Escherichia coli is this palindromic sequence, GAATTC. And when EcoR1, the restriction enzyme, is allowed to digest DNA, it doesn't just go through and cut the piece of DNA inadvertently. It attaches to the DNA molecule and it travels along the DNA molecule until it finds this recognition site, and it cuts the sugar phosphate backbone between the A and G nucleotides, which destabilizes the molecule and causes the hydrogen bonds between the A's and the T's to break and opens up the DNA molecule. So these restriction enzymes are said to digest DNA, and when they digest DNA, what we mean by that is that they are moving physically along a piece of DNA, and everywhere they find this restriction sequence or restriction site, they cut the sugar phosphate backbone, thereby opening up the piece of DNA. And the really cool thing about EcoR1 and a couple of these other enzymes is that when they cut the sugar phosphate backbone of the DNA, they create these single-stranded fragments, which, for instance, with EcoR1 is this AATT. And that's cool because that AATT single-stranded piece of DNA will be drawn to and want to attach to any complementary sequence, because that A will want to bind to a T, and the A next to it will want to bind to a T, and the T next to that will want to bind next to an A, as will that final T. And because that AATT single-stranded sequence wants to bind its complement, is called sticky. And that stickiness of the DNA is what enables us to combine molecules of DNA from two sources. All right, in this particular example, you have a restriction site on the plasmid, GGATCC, which is the restriction site not of EcoR1, but of BAMH1. So this is a different restriction enzyme. And this restriction enzyme, regardless of the site, still has the ability to create those sticky ends because it breaks the sugar phosphate backbone between those adjacent guanine nucleotides. So when you digest this plasmid with BAMH1, you will create a linear piece of DNA which has these sticky ends. So in a separate test tube, you take the cellular DNA, right? In this case, it would have been the human genome, and you use the same restriction enzyme to digest the cellular DNA, and then you isolate out the piece that has the insulin gene within it. So now that little red piece of DNA, the portion that is not read out is the insulin gene. So now you have your insulin gene isolated, and you have in a separate test tube your plasmid, which has been opened up, so now it's sticky and wants to attach to that. And when you put those two things together, they can anneal to one another, they can attach to one another, they can attach at those sticky ends, because the GATC portion of the human DNA is exactly the same and is complementary to the CTAG portion of the plasmid. So not in all of the plasmids, but in some of the plasmids, you get this recombinant DNA. You get a piece of DNA that has that insulin protein within it, that has the gene of interest. As I mentioned, there is such a wide variety of biotechnological applications that we're only going to talk about a few of them. The first was restriction enzymes. The second is something called polymerase chain reaction. And the reason that we chose to look at these is because the restriction enzymes are actually enzymes isolated from bacteria, and the polymerase chain reaction is a way of cloning or making copies of DNA in vitro that relies on a DNA polymerase that has been isolated from an extremophile, which, as you know from previous lectures, is going to be a prokaryote. So talking generally about DNA replication, we know that you have to do two things. Number one, you have to expose the nucleotide bases because they exist inside of that DNA molecule, and then you have to replicate the DNA. In vivo, in the cell, the normal process is that the genome serves as a template DNA. We know from Messelson and Stahl that the DNA molecule opens up and new nucleotides are added opposite their complements in the parental strand. So you need that template DNA, you need the nucleotides to add to the daughter strands of DNA, and you need, among other things, three very important enzymes. One is DNA helicase, which actually opens up and exposes the nucleotide bases of the DNA. You need that very important DNA-dependent RNA polymerase or DNA primase, which provides the 3' OH group to which the final enzyme DNA polymerase will attach and actually add the nucleotides to the growing fragment of DNA. So if you want to make this process happen in vitro, you've got to figure out a way to mimic these natural conditions. So first, you'll need a template piece of DNA, which can be any piece of DNA that you choose. You need nucleotides to create the new piece of DNA. In order to not have to use DNA helicase, you can instead use heat to denature the DNA to single strands, and you can use short primers instead of DNA primase in order to direct the synthesis of DNA to the portion of that genome that you would like to create copies of. And because you're using heat, you need to use a DNA polymerase that is thermostable. And that's where the microorganisms become important, because any DNA polymerase that is thermostable has to have come from a thermophilic organism. And there are two reasons that the DNA polymerase has to be thermostable. Number one is the fact that you have to get DNA to near boiling temperatures in order to denature it. The second is that you utilize heat to make sure that the primers bind only where they are complementary to the template DNA. Imagine you have the human genome, 3 billion base pairs, and you want to amplify or copy or clone the gene for insulin. You have to have a way of directing the DNA polymerase to that insulin gene, and you do that by providing primers. Primers are short, single-stranded pieces of DNA which are complementary to the outside edges of your gene of interest. So primers will allow you to direct the area of the chromosome that you're going to be replicating, because if they don't bind or anneal to the DNA, the 3' OH group will not be present, and the DNA polymerase, even if it's thermostable, will not be able to produce that complementary copy of that DNA strand. So during the PCR process, we're going to see that there are actually 3 temperatures involved. There's the denaturing temperature, which actually opens up the DNA molecule, and then there's an annealing temperature, and then there's an elongation temperature. The elongation temperature is the temperature at which DNA polymerase is active, which as a thermophile is usually around 75 degrees Celsius. So 75 degrees Celsius is enough heat that if those base pairs don't match, they won't anneal. As you see with the top example here, there's not enough complementarity between those 2 fragments of DNA, and so there's no annealing, and that means that there's no OH group there for the DNA polymerase to bind to. As opposed to the bottom example, which, because it's perfectly complementary to the template DNA, can withstand that 72 degrees Celsius. And so when DNA polymerase comes along, it will recognize that OH group and start to replicate the rest of that piece of DNA, utilizing that OH group to catalyze the reaction. So polymerase chain reaction has cycles that are made up of 3 temperature profiles. And each cycle begins with a denaturation step, which is about 95 degrees Celsius, which will denature the DNA into 2 single strands, exposing the nucleotides. Then it will drop down to about 50 degrees, maybe up to about 65 degrees to allow those primers to anneal. And then the temperature will increase to 72 degrees, which will test the fidelity of the primer template binding and activate DNA polymerase to copy the pieces of DNA only where those primers are bound. And so at the end of that cycle, you have exponential growth of the DNA molecule. You had 1, and now you have 2. And then you just keep cycling that process over and over and over again until you have billions of copies of DNA, much like in exponential growth, binary fission allows for a very rapid doubling of the microbial cells. Once you have DNA molecules, whether you have created them and you just want to look at the entire molecule, or if you've created them and you digested them with restriction enzymes and you want to look at them, you need a way of separating and visualizing those fragments based on their size, and that can be accomplished utilizing something called agarose gel electrophoresis, which utilizes electricity to move DNA samples through a thick gel matrix. And the electricity is used because DNA is a negatively charged molecule, so you can place a negative charge at the top of the tray and a positive charge at the bottom of the tray, and the DNA will be repelled by the negative and drawn toward the positive charge. Because you're making the DNA move through this gel matrix, larger pieces will be impeded as they try to move through the gel, so over time, you can create these banding patterns of DNA based on their size, and you can use those for multiple different applications. We can actually use PCR in order to identify pathogenic genes, and that was done in this set of experiments here, where a bunch of patients were colonized by E. coli, and they were trying to figure out whether or not the E. coli that was colonizing these patients was actually the pathogen causing their disease. And it turns out that the difference between E. coli, the normal flora strain, and these pathogenic strains of E. coli is the presence of different pathogenic genes. So you can have primers that will anneal to the sides of these pathogenic genes and create copies of those pathogenic genes, but only in the strains which have those genes. So it's a way for you to look at the genotype in order to identify a phenotype in the patient. So on the very left-hand side, we have a negative control. This is just a PCR with no E. coli whatsoever. In what is labeled lane 3, we have an enteropathogenic E. coli strain. So you see one band there that indicates a pathogenic gene. Next to him in lane 4, you have an enterotoxigenic E. coli, which has 2 fragments of DNA that are associated with pathogenic genes. And then finally, enteroinvasive E. coli is shown in lane 5 with one other distinct and recognizable pathogenic gene, and then that is compared in lane 6 to E. coli, which are part of the normal flora. So all of these patients, if you did biochemical testing, would test positive for E. coli. So now we need to know whether these patients on the right-hand side of this agarose gel electrophoresis data are actually sick because of those E. coli strains. So patient 1 has a band that lines up perfectly with enteropathogenic E. coli, so that patient has this pathogenic strain of E. coli. Patient 2 and patient 3 have none of the genes that are associated with pathogenic E. coli. Patient 4 actually has a banding pattern that was not tested for in the initial experiment, but lines up with a 4th pathogenic strain of E. coli called verocytotoxigenic E. coli. Patient 5 does not have a pathogenic strain of E. coli. Patient 6 has the enteroinvasive E. coli, and patient 7 has enterotoxigenic E. coli. So PCR just of pathogenic genes can very quickly differentiate a normal strain of E. coli and pathogenic strains of E. coli, and it can do that for other disease-causing organisms as well, because many organisms cause disease only when particular genes that create toxins are present. Notice there was toxigenic, verotoxigenic, enterotoxigenic. These are all genes that produce toxins that are themselves the cause for the disease symptoms in the patient. I would like to talk about just a couple of topics where microorganisms and the study of microbiology has had far-reaching effects in industrial or commercial applications. One of the ways that microorganisms very commonly cause disease is through something called oral fecal transmission. So whenever you contract something by eating or drinking contaminated water, you have been inoculated with what likely came at some point from the feces or stool of an infected person or animal. So that might be something that's on the hands of that person and then gets transmitted to their own mouths via auto-infection. It might be that that person's hands are contaminated and they touch a foam mite, an inanimate object, which then touches your food like that plate. It can be that vectors like flies or other arthropods will land on the fecal material and transmit that to you. It can be transmitted through the soil. It can be transmitted in water. And there is lots of applied microbiology going to trying to stop oral fecal transmission, either because of toilet barriers or water barriers, and then what you can do yourself to protect you from these orally fecally transmitted diseases is hygiene and hand washing. So we're going to talk about this idea of oral fecal transmission much more in our last lecture unit, but here we're going to talk about how we can use microbiology to identify possible sources of oral fecal contamination of water. What you need to know in terms of microbial water pollution is there fecal organism growth there? And the way that we determine that is by looking for the presence of what are called indicator organisms. Indicator organisms, as their name suggests, indicate the presence of fecal contamination. And the best indicator organism that we have available is Escherichia coli because it's ubiquitous in the guts of human beings and most other mammals, and it can't survive for very long outside of the body. So if you find E. coli, then you have found evidence that within maybe the last week or so, there has been fecal contamination of that area. And you might remember from the PCR reactions that I just showed you a few minutes ago that not all E. coli are pathogenic. So it doesn't mean that that E. coli is causing or is in danger of causing illness. It's an indicator that fecal contamination is present, and where fecal contamination is present and where E. coli are present, there's a very high likelihood that other more difficult to find or grow fecal contaminants are there. And in order to look for E. coli, you can do something called a mug coliform test, which is actually something that you will get to perform on a water sample of your choice. So the mug ColAlert test is a commercially available kit that has two reagents or substrates in it. One is ONPG, and you don't need to worry about what that stands for. The other is MUG, or MUG. And these two chemicals are important for two reasons. Number one, they're capable of inducing two different bacterial operons. And the other important thing is that they change color in response to that induction. So the color change will indicate to you that organisms are present. Now, ONPG, which induces the lac operon, will induce that operon in any organism that has the lac operon, and that includes lots of organisms that are packed together into what we call the coliforms, the gram-negative bacilli. So getting ONPG, which is converted by beta-galactosidase, which is part of the lac operon, to turn to a yellow color, simply indicates to you that there are coliforms present, which, if you're talking about a recreational water sample, may be part of the normal flora. However, MUG, which induces the glucuronidase operon, will typically only be induced by E. coli because this is a much less utilized operon. The production of beta-glucuronidase is indicative of the presence of E. coli, and so the conversion of MUG by beta-glucuronidase into this fluorescent compound, which you see in the photograph here, is indicative of E. coli, And so if you were talking about drinking water, you wouldn't want to find a yellow color change, which would indicate coliforms, or a fluorescent color change, which would indicate E. coli, because drinking water should be free of microorganisms. If instead you're talking about recreational water, a lake, a stream, the ocean, something like that, coliforms would not necessarily be an indicator of anything dangerous in the environment, whereas fluorescence caused by E. coli would be an indicator of fecal contamination. So this allows you to be far more precise in what you're saying about a given water sample, not just something's growing, but something's growing that is a coliform, or something's growing that is E. coli. And when you have E. coli growing, you have very likely fecal contamination. Continuing on this vein of illness as a result of contaminated food, we want to talk for a moment about food preservation, which we have talked about in previous lectures. When we talked about the control of microbial growth, we talked about causing osmotic stress to bacterial cells by drying them, foods that is, by drying food or salting food or sugaring food such that it causes osmotic pressure. All the water is drawn out of the cells and they're not able to grow on the food, which preserves it. We also talked about fermentation in previous lectures, where microorganisms will ferment, and oftentimes the products of that fermentation, the ferments, are noxious or toxic to the microorganisms. And so they preserve the food. And in addition, we talked about the fact that a lot of times, right, fermentation is a benefit not only because it preserves the food, but actually imparts different flavors to the food. So those are historic methods of food preservation. More recently, the idea of sterilizing and aseptic packaging, along with some of these newer technologies that we talk about in our control of microbial growth lecture, have led to foods that are sterile and have a shelf life of multiple years, right? And one common is canning, which can be done at home or can be done industrially, as is shown here, right? Food is cooked, it's put into a container, and that container is sterilized and sealed. And so those products can be stored at room temperature for years. That also can be done after foods are irradiated or exposed to high energy electrons or high pressure. Any of those things will sterilize the food, and then it can be aseptically packaged into sterile containers in order to preserve the foods for much, much longer than they would be naturally safe to consume. Now, as we mentioned, this is not a new concept, and in fact, Louis Pasteur came up with something that he called pasteurization in the 1800s, where he was actually heating wine at 60 degrees in order to kill pathogenic bacteria and deactivate spoilage enzymes. And when he published those works, as you can see here in 1876, that was picked up by Franz von Soxhlet, who applied this idea of pasteurization to milk and came up with this modern idea of pasteurization, where milk is exposed for 20 minutes to 60 degrees Celsius in order to reduce the number of spoilage organisms. Now, milk exposed to historic pasteurization still has to be refrigerated because it still has organisms in it. It's not sterile, but it will last for much longer than it would otherwise. Today, our milk is exposed to what we learned in our earlier unit. It's called ultra-high temperature pasteurization, exposure to 135 degrees Celsius for 2 to 5 seconds, and that allows the milk to be pasteurized, but to maintain more of its natural flavors. And this method of pasteurization, though it doesn't sterilize the food, eliminates Listeria and Salmonella, Staph aureus, Yersinia, Campylobacter species, enterohemorrhagic E. coli strains, and so it prevents a lot of disease transmission in addition to the fact that it provides a longer shelf life. In addition, the same recombinant DNA technology that we talked about at the beginning of this lecture can be used not for pharmaceutical products, but for industrial products, where genes of interest from some industrially useful organism are integrated into bacteria through the process of transformation, and then the genes from those can be inserted into plants. They can be used for bioremediation, they can be used for other industrial applications, and they can be used to treat patients. James Curry, who worked along with a 16-year-old assistant named Jasper Kane to up the production of penicillin after its discovery in the 1920s so that it could be used in order to protect Allied troops during World War II. And the problem with penicillin is it's finicky, it's difficult to grow, and even when you produce penicillin, there's about a 1 to 10,000 ratio of penicillin to the broth. So you have to have a ton of broth, a large-scale production, in order to make enough penicillin to be therapeutically useful. But if you have these giant vats, the problem is that this mold is an aerobe, and the bottom of the vat would not support the growth of that aerobe. So Curry and Kane devised 14 75,000-gallon tanks, and they used them in 1943 to start producing penicillin. And it was really exciting because right off the bat, they produced 5 times the penicillin that they thought they would be able to. And on D-Day, every Allied troop had a dose of penicillin, and 90% of those penicillin doses were made by Pfizer through the work of these two men, James Curry and Jasper Kane. And that work was done utilizing the deep tank fermentation technology that Curry had started working with in Pfizer to make citric acid. And these deep tank fermenters, or bioreactors as they're sometimes called, are basically enclosed containers where every conceivable aspect of the environment is controlled. If you get too acidic, a base will automatically get added to raise the pH. If it gets too alkaline, an acid will be added to lower the pH. If it gets too warm, then a cooling agent will be added to the outside to cool it down. If it gets too cool, it'll get heated up. It's aerated, and it allows for the organisms inside it to grow to stationary phase, and instead of moving into death phase because of the buildup of toxins or the lack of nutrients or the lack of space, those organisms are capable of staying in stationary phase. And that allows for a maximal amount of whatever you're looking for in this fermentation product to be harvested. So when we're talking about deep tank fermentation and bioreactors, notice we're not talking about lag and exponential and stationary and death phase. What we want to talk about is cells that are at a low concentration and cells that are at a high concentration. And so those are differentiated by trophophase and idiophase. So trophophase is cells anywhere before stationary phase. Idiophase is where the cells enter stationary phase. And the reason why this idiophase idea is so important is because many things will not be produced unless the cells are at high concentration, which may sound familiar to you, right? That's quorum sensing. So for instance, penicillin. Organisms produce penicillin to kill off anybody that's competing with space for them. So they don't need to produce penicillin until it gets crowded. So they use those interspecies chemical cues and intraspecies chemical cues from around them. And when those chemical cues get to a high enough concentration, they realize that the cell density is high enough that they need to start killing off the competition, and so they produce penicillin only during idiophase. So that means that if we want to harvest penicillin, we need to get the cells to idiophase and keep the cells at idiophase for as long as possible so that we can extract as much penicillin as possible. Now, even if you have something that's produced all the time, say ethanol, which is another commercially useful product, you still have a vested interest in getting the cells to idiophase because the more cells there are, the more ethanol they will produce. So whether you are producing something that is what's known as a primary metabolite, something that is produced no matter where the cells are in this cycle of growth, or a secondary metabolite that is not produced until the cells enter the idiophase portion of growth, you will want to have them growing in deep take fermentation bioreactors so that you get the maximal product necessary. Now, as I mentioned, the industrial products of deep tank fermentation and other things are so varied that it really would be difficult to mention all of them. But just for instance, amino acids are produced, proteins are produced, vitamins, steroids, antibiotics, as we just saw, drugs, ethanol, as we just saw, methane gas, all sorts of different things. And they're all made because bacteria have been found that do those things naturally, and then that natural ability has been exploited in vitro in order to accomplish a particular task, which is either pharmaceutically or industrially important.